As of today, cancer is a disease that is becoming more common. This may be caused by a tumor suppressor, gene mutation, cell-induced hypoxia, or the expression of VEGF in a cell. First, I will give you the background information that you need to know. Proteins are one of the 4 macromolecules. In order for proteins to be synthesized, a strand of mRNA must be made from DNA. Once the mRNA is made, it leaves the nucleus through a nuclear pore and into the cytoplasm. The mRNA will travel to a free ribosome or a ribosome on the rough endoplasmic reticulum. The mRNA runs through the ribosome and amino acids are ready to bind to create a protein. Amino acids link together by the formation of peptide bonds to make proteins. Protein turnover is a system of proportion between protein synthesis and protein degradation. Proteins have different shapes. The simplest structure is a linear assembly, which is the primary structure. For secondary structures, an alpha helix or beta pleated sheet can be formed. Hydrogen bonds form to help maintain the shape of the protein. These secondary structures combine in different arrangements to create a tertiary structure, and disulfide bridges form to hold those secondary structures to one another. Quaternary structures form from the assembly of many tertiary structures. These structures can be denatured through an unfavorable cell environment, such as a high or low pH or temperature. The protein can, but will not always have the capability to reform into its original shape. Proteins can be degraded by either autophagy or the ubiquitin proteasome system. In the process of autophagy, a vesicle surrounds damaged material and organelles. The lysosome engulfs the vesicle to break down its components with hydrolytic enzymes. Autophagy is later signaled to no longer perform its function by suppressing the tumor. A suppression of autophagy is not good for the cell because this process protects the cell from foreign invaders such as cancer. In the ubiquitin proteasome system, or UPS, a protein called ubiquitin marks damaged or denatured proteins. When a protein has 4 ubiquitin markers, it is known to be polyubiquitinated. It will then move on to the 26S proteasome to be cut up into amino acids. Hypoxia-inducible factor, or also known as HIF-1-alpha, is a protein that is related to cancer development when the cell is in a low oxygen environment. The VHL, which is a tumor suppressor, carries ubiquitin and looks for a specific hydroxyl group on HIF-1-alpha. However, in hypoxic conditions, the specific hydroxyl group isn't present. This will result in the expression of VEGF, a growth factor, which is also found to be present in high levels in cancerous cells. The expression of VEGF promotes the growth of the tumor, increases HIF-1-alpha levels, and promotes the grouping of HIF-1-alpha. Oncoproteins promote the growth of a tumor, and it affects how HIF-1-alpha will react in the cell. The tumor prevents apoptosis from occurring. Apoptosis is when the cell leads itself to death. According to Ayano, the HIF-1-alpha affects the tumor aggressiveness, invasiveness, and resistance to radiotherapy and chemotherapy. Since chemotherapy will not greatly impact the tumor, a way to treat patients is through the inhibition of the proteasome. Bortezomib is a well-known drug to treat cancer, specifically refractory multiple myeloma. When the drug takes effect in the patient, the cancerous cells are signaled to undergo apoptosis.